All right, so for today, what we're going to be is we are going to be taking a look at a couple processes called transcription and translation. So again, we're a little bit ahead of our lecture. So this is going to be a process we're going to talk about in more detail next week in lecture. But here we're going to be looking at a couple processes called transcription and translation. Okay, so in the past, we've been looking at DNA and how DNA is built, how it's going to, or we haven't talked about how it's built, but we're going to be looking, or we have looked at how it's able to replicate to help us be able to produce new cells. We've looked at how it's able to help us produce new individuals that are unique to the parents and to the siblings. And so today what we're going to be doing is we're going to be looking at how we can actually activate specific genes to produce the different proteins and characteristics that we want. Okay. So that's going to be what we're doing today. Okay, so when we were first talking about DNA, one of the things that we said was that DNA is basically like a very large instruction manual. Okay? It's a very large instruction manual to help us produce various types of proteins. Okay, the problem with DNA is it's kind of like a very, very thick, dense instruction manual. Okay, and there's a lot of information in there that doesn't necessarily need to be there. Okay, and so it's kind of got a lot of like extraneous information, things that we don't necessarily need at that point. Okay. So what we want to do is we want to take DNA, we want to take that very large instruction manual, and we want to shorten it. Okay, we want to make it more concise so that it has just the information we need. Okay. So in order to do that, what we're going to do is we're going to take our DNA here, and we're going to transcribe it into a more concise, shorter RNA molecule, or also called mRNA, or messenger RNA. Okay. Now that we have a more concise instruction manual in our RNA, okay, we're then going to use that information in there. We're going to translate it to help us form our protein. Okay, and so this is called the central dogma of biology. This is going to be a central theme in biology that everything is going to kind of revolve around. Okay, we're going to start with some DNA. Okay? Our DNA is eventually going to get transcribed into RNA, or messenger RNA, and then from there, that RNA is going to be used and translated to help us form some proteins. Okay, and then these proteins will be able to perform various functions throughout the cell and throughout our bodies here. And so these two processes are going to be taking place inside of the cell. The first process is transcription, where we're going to be taking our DNA to help us form some mRNA. Okay? And so this is going to be a process that takes place inside the nucleus. Okay? So transcription happens inside the nucleus where we form our mRNA molecule. Okay? Now, once we form that mRNA, okay, the next thing that we're going to do is we need to remove that mRNA from the nucleus. Okay, so we're going to just spit it right out of the nucleus and into the cytoplasm. Once it enters the cytoplasm, we can then perform translation, where we use that mRNA to help us produce a protein. Okay? And then again, that protein will be able to perform various other functions throughout the cell. Okay, so that's the game plan for today. What we want to do is we want to look at how we can start with a DNA molecule and how we're going to end up forming a protein. Okay? That's our goal. Okay. Now, before we can do that, we're going to kind of start at the beginning of this whole process here, and we're going to look at DNA. Okay? And so if we remember from like high school biology, DNA is organized as kind of a double helix, meaning that it has two strands that are going to be oriented around each other and kind of wound around one another. Okay? So we're going to have two strands of DNA all wound together here. Okay? Now, DNA in and of itself cannot just kind of exist as two strands, but it has to be held together by something. Okay? So if we unwind that DNA, what we're going to do is we're going to see that we kind of see these kind of a ladder formation here. And we're going to see some nucleotide bases, okay, that are going to hold our two strands of DNA together. Okay. Those nucleotide bases, okay, there's a total of 4 of them, okay, are adenine, adenosine, okay, which is A. We have thymines, which are our T's. We have cytosines, which are our C's, and then G, which is our guanine. So our rungs of our ladder, okay, there are our steps here. They're not just going to be, held together any which way. There's a very specific manner in which we're going to be holding our DNA strands together with. Okay? So what we're going to do is we're going to have our A's, our adenines or adenosines. Those are always going to be bound to the thymines or our T's. Okay, so A's and T's will always be bound together. The guanines will always be bound to the cytosines. Okay, so our G's are always going to be bound to our C's. Okay. So that's how the formation is always going to be. Okay, the adenines, or so the adenosines on one strand are always going to bind to the thymines on the opposite strand. Okay, and the cytosines on one strand are always going to bind to the guanines on the other strand. Okay, so A's will always bind with T's, C's will always bind with G's. Okay, always, always, always. That's going to be the same formation we'll always see in our DNA. Okay, and so this is what holds our DNA together. Okay, these nucleotide bases here. Okay, so there's a rule that we're going to see mainly next week. That tells us that the amount of adenosines that we see, the amount of A's is always going to be equal to the amount of T's that we have, which makes sense because our A's are always going to be binding with the T's. Okay, so the amount of A's we have are always going to be the same as the number of T's. The amount of C's that we have are always going to be the same as the amount of G's. Okay, so adenosines will always bind to thymines. Cytosines will always bind to guanines. Okay, A's with T's, C's with G's, always, always, always. Okay, so for that reason, we will call these, or we always classify these as complementary base pairs. Okay, they're complementary bases because obviously these are nucleotide bases, and they're going to be complementary because they will always go hand in hand with each other. Okay, A's always with T's, and C's always with G's. Okay, so this makes sense so far. This is kind of going to be an important thing to help us get straight before we go forward here. Any questions on this? Does this make sense? Okay. All right, perfect. Okay, so a quick example here. So we have a DNA sequence on one strand is as follows. Okay, we have A, T, G, C, A, G, T, A. Okay, that's the DNA sequence on one of our strands. Okay, so what will be the complementary sequence on the opposite strand of DNA? Okay, so we're going to take a look at these and see if you can figure out which of these is going to be the complementary strand or the complementary sequence to this sequence up here. What do we think? Right, C is going to be the answer here. Perfect, good job. So this strand, C, is going to be our complementary strand to the strand up here. So here we have an A, so its complementary base would be the T. Here we have a T, so its complementary base would be the A. Okay, G, so its complementary would be the C. C, so we'll have a G. A, so we'll have a T. G, so we'll have a C. T, so we'll have an A, and then an A, so we'll have a T here. Okay, so that's going to be our complementary bases. They're always going to be exactly the opposite of one another. Perfect. Okay, so that we've kind of cleared that part up. Now we're going to get into transcription. Okay, so transcription is going to be the process of converting a DNA molecule and helping us form a messenger RNA molecule or an mRNA. Okay. Now in prokaryotes and eukaryotes, this process is going to look a little bit similar, but it's going to be taking place in different locations. Okay. So in eukaryotes, transcription is going to occur only in the nucleus. Okay. So we're going to be forming that mRNA inside the nucleus, and then from there, the mRNA is going to get transported into the cytoplasm, okay, in order to undergo translation later on. Okay. But we're going to be performing this process inside the nucleus for eukaryotes. Okay. Now, in the case of prokaryotes, prokaryotes do not have a nucleus, so their transcription process is going to take place just inside the cytoplasm. Okay. So when we look at transcription, transcription is going to take place in 3 stages. Okay. There's going to be an initiation stage, then we'll have an elongation stage, and then the last thing we'll have is a termination stage. Okay. So those are our 3 stages. So the first stage that we're going to have is the transcription stage. Okay. So, sorry, is the initiation stage. Okay. So initiation, the first thing that we need to do is we need to take our DNA double helix and we need to unwind it. Okay. So now it's in kind of our ladder formation. Okay. So here we have one strand on one side and then the other strand on the other side. Okay. So we're going to unwind our DNA strands. Okay. And then we're going to kind of partially pull them apart. Okay. Pull apart a part of our DNA as we see over here. Okay. And so this is what's going to form what's called our transcription bubble. Okay. So this is where we see our transcription bubble here. Okay. We see a little bulge over here and a little bulge over here where we start to pull apart our DNA. Okay. The next thing that's going to happen is we're going to have a bunch of proteins and enzymes, okay, that are going to bind to a specific region on the DNA called the promoter. Okay. So the promoter is in front of the gene. Okay. It's upstream of the gene that you're going to activate. Okay. That promoter is going to tell you where exactly transcription is going to begin. Okay. So every single gene is going to have its own promoter sequence, and And so again, that's going to tell you where you're going to begin transcription. Okay. So all these enzymes are going to bind to the transcription or to the promoter, which is going to be upstream of that particular gene that you're ready to activate. Okay. So that's the initiation stage. Okay. Again, we're just unwinding our DNA, forming the transcription bubble, and then we have a bunch of our enzymes beginning to bind to the promoter. Okay, now the next thing that's going to happen is we are going to start the elongation phase. Okay, so the elongation process is going to be where we start to actually make the mRNA. Okay, now in order to do so, we're going to be starting from one of our DNA strands. Okay, one of our DNA strands is going to be used to help us make the mRNA molecule. Okay, we call the DNA strand that we're going to be using the template strand. Okay, so we're going to have a template strand that's going to be used to help us make our mRNA. And then we have a non-template strand that's not going to be involved with this process. Okay, so in order to make the mRNA, we're going to be using the template strand of DNA. Okay, now when we form our RNA, okay, we're going to be adding RNA using our RNA nucleotides using an enzyme called RNA polymerase. Okay, so RNA polymerase is going to be what helps us actually make the RNA. Okay, now when it does that, it's going to be adding nucleotides in a complementary manner to the template strand. But it's going to be adding those nucleotides to the 3' end of the RNA. Okay, so we'll talk more specifically about the 5' and 3' end, but essentially when we're looking at each strand of DNA or RNA, there's going to be a 5' end and there's going to be a 3' end. We'll talk about what that means next week in lecture, but essentially just to orient you guys, one DNA strand is going to have a 5' end on one side. Okay, so that's our 5' end. The other end is the 3' end. Okay. The opposite strand will have the 5' end adjacent to that. Okay, and then this opposite side will have the 3' end. Okay, so those are the 5' and 3' ends. Okay, so when we're adding our RNA nucleotides, we're going to be adding them in a complementary manner to the template strand. Okay, but we're going to be adding those nucleotides from the 5' end to the 3' end. So we're going to be adding these nucleotides onto the 3' end of our RNA here. Okay. So we're always going to be adding to this side. Okay. So let me go ahead and move forward here so we can have another way of looking at this here. So when we're looking at our RNA molecule that we're going to be producing, okay, we're going to have the same nucleotides, okay, with the exception of one. Okay, we're still going to have our adenines or adenosines. We're still going to have our cytosines. We're still going to have our guanines. So we still have A, C's, and G's. However, in RNA, we are not going to have any T's. Okay, we're not having any thymines. In RNA does not contain any thymines, and instead it replaces thymines with another nucleotide called uracil, which we'll put the U for that region here. Okay. So again, when we're forming the mRNA, okay, the mRNA that we produce is going to be complementary to the template strand. Okay. So here's our template strand. Here we have a T. Okay. So the RNA is going to form an A. Okay. Here in the template strand we have an A. Okay. So its complementary base would be a T, but here because we're looking at at RNA, okay, we're going to have a U instead. Okay. So its complementary base in RNA would be a U. Okay, for A's. Okay. Then we have a C. Okay. So our complementary base would be G. Here we have a T, so its complementary base would be an A. Here we have an A, so its complementary base in RNA would be a U and not a T, okay, because we don't have any T's. Okay. And here we have a G, so its complementary base would be a C. Okay. So does this make sense so far? Again, we're still dealing with the same complementary bases here. The only difference here is that we have U's instead of T's. Okay. All right, so let's go ahead and continue adding to this strand here. So I'll go ahead and put one here. Okay, so let's go ahead and continue adding to this. Okay, so let's say, so here we have our A. Okay, so what would its complementary base here be? What should we have? So not a T, it wouldn't be a T because again, we're looking at RNA. RNA does not have any T's and we replace them with what? A U, exactly. So we're gonna have a U here. So our A's are going to have a complementary base for the U here. Okay. Next, we have a G. So what's its complementary base going to be for RNA? C. Perfect. Whoops. C. All right. Then we have a C here. So what should we have in our RNA? G. Perfect. Then we have an A here. So what should we have in our RNA? Anyone? A U. Perfect. Good. Then we have a T here. So what should we have here in the RNA? An A. Perfect. And then our last one, we have another T. So we should have a what? An A. Perfect. Good. So here we've just completed our RNA. We've completed building our RNA, right? So we had our A's binding to T's, C's binding to G's. However, again, when we're looking at RNA, RNA does not contain any T's. All it has instead of T's are U's. Okay? But everything else is going to be the same. Okay, we still have A's, we still have C's, we still have G's. Okay, but instead of having T's, we're going to have U's. Okay. Okay. So the mRNA, like we said, is going to be complementary to the template strand. Okay. Now, another thing to keep in mind here is that when we're looking at the non-template strand, or also called the coding strand and the RNA are both going to look very, very similar. Okay, almost identical. Okay, which makes sense because the non-template strand should be complementary to the template strand, right? And the RNA is also complementary to the template strand. So if they're both complementary to the template strand, then the non-template strand and the RNA should look almost identical. Okay. The only thing that's different here is that we're going to have U's instead of T's in the RNA. Okay. So again, here we have A's, U, then G here, A here, then we have a U here, then C's, we have a U, C's, G, U, A. Okay. So the non-template strand or the coding strand is going to look very identical to the RNA, and both of them are going to be complementary to the template strand. Does this make sense so far? Any questions on this process here? Okay, so that being said, there's a couple tricks to this. And one thing that I'll bring up mostly in lecture is that, for example, if you know the template strand, you can identify what the RNA strand is going to be because, again, you're just going to use the complementary bases. But if for whatever reason you don't have the template strand and all you know is the non-template strand, okay, you can still identify what the RNA is going to be because it's going to be identical. Okay, the only difference again is going to be that you're going to have U's instead of T's. Cool. Okay, so that's our elongation process. Okay, so that's how we're going to form the mRNA. Okay, and again, we're going to be adding that to the 3' direction of the RNA molecule. Okay, cool. Okay, so when we're completing elongation, or I guess when we're going through this process here, the RNA that we produce is initially going to be bound to the DNA at the template strand, or the DNA nucleotides at the template strand. Okay. Now, eventually, that RNA is going to unbind from it, and then it's going to kind of be its own thing here. It's going to have its own individual structure. Okay. So it's not going to remain bound to DNA. Okay. Now, RNA polymerase is going to keep on moving. Okay. It's going to keep on moving and keep adding these RNA nucleotides in a complementary manner to the template strand. Okay. As it continues moving, okay, the DNA ahead of RNA polymerase is going to be unwound, okay, in order to help access the template strand. Okay. And then as RNA polymerase moves out of our particular region, the DNA behind it is going to get rewound back up into its original formation. Okay. All right, so that's elongation. And that's pretty much how we're going to end up forming our RNA molecules. Okay. So now we're going to go ahead and do a quick activity here. So let me know when you guys are able to see our transcription activity. Okay. Is everyone able to see it? Yeah, okay. Okay, cool. So here we are looking at a nucleus. Okay? And we've got a bunch of DNA all packed within this nucleus. Okay? So we want to transcribe some of our genes here. Okay, so here we've got 3 different genes, 1, 2, and 3 here. Okay, we're going to choose one of these genes that we want to activate. So I'm just going to go ahead and activate this one. Okay, we're going to choose a gene. Okay. I'm going to skip through this video here. And so this should get going relatively soon, hopefully. Any second now. Any second now. Okay, okay, here we go. All right, so here is the DNA that we are ready to begin transcribing. Now, whenever this thing wants to start, we'll eventually start transcribing our gene. But this thing is taking a little while. Okay, this wasn't supposed to happen. Okay, so I'm gonna go ahead and try refreshing this, and then we'll see if it wants to cooperate this time. Okay, here we go. Oh, what happened? Okay, gene. Okay. All right, sorry about this. Almost ready here. Okay, here we go. Okay. All right, so now we have our gene that we're ready to transcribe. Okay. And so here we have our DNA sequence. Okay. So this is our DNA sequence, and we need to add the correct RNA nucleotide. Okay. So if we have a T here, what is going to be the correct RNA nucleotide? It's going to be C, A, G, or U. A, exactly. Perfect. So we're going to add an A there. Okay. Here we have another T. So again, what should our RNA nucleotide be? Perfect. Another A. Good. Okay. So now here we have an A in our DNA. So what should the correct RNA nucleotide be here? U. Perfect. Good, Jackie. So we should have a U there. Cool. Now we have a C. So what's the corresponding nucleotide? G. Perfect. Brianna, good job, Brian. Good. Okay. Then we have an A. So what should we have? We should have a U. Perfect. Okay. Now we have a G. So perfect. We should have a C. Good. And so as you can see, we're starting to produce our RNA here. Okay. What about G? Another G. Here we should have, perfect, another C, good, okay. Here we have an A, so what should we have? U, perfect. Then we have a T. So what do we need? An A, good. Okay. And we have a C. So what are we looking for? A G, perfect. Okay, here we have a T. So we need an A, good. Okay. Now we have a G. So what do we need? A C, perfect, good job. Here we have a T again. Okay, so what do we need here? Perfect, we're gonna need an A nucleotide. Okay, here we have a C nucleotide. So what nucleotide do we need here? Right, we're gonna need a G, perfect. Okay, here we have an A. So what do we need? U's, perfect. All right, so at this point, we've just formed our RNA. Okay, so now, so that's like the RNA that we just formed through transcription. Okay. So now, let me go ahead and pause this real quick. We'll go ahead and come back to our PowerPoint here. Is everyone able to see the PowerPoint again? Yeah, okay. So we've just completed transcription. We've just formed our mRNA molecule here. Okay. So now that's going to bring us into the next process called translation. Okay. So what we need to do now is we need to take that mRNA that we just produced from transcription, and we need to use that to help us form some proteins. Okay. So translation is basically protein synthesis using an mRNA molecule as kind of our shortened instruction manual here. Okay. And so in order to produce a protein, remember that proteins are going to be made up of a bunch of amino acids that are all linked together. Okay. So in order to build our protein, what we need to do is we need to string one amino acid to the next one using covalent bonds, and that will help us form a very long string of polypeptides and eventually proteins here. Okay. So we're going to be adding individual amino acids one at a time, okay, to help us produce the polypeptides and proteins that we need. Okay. And then from there, those proteins will be able to go off throughout the cell and be able to perform various types of functions, okay, depending on the protein in question. Okay. So before we get into translation, we got to kind of introduce ourselves to the big players here for this process. Okay. So the first most important structure that we're going to need for translation is the mRNA template that we just produced from transcription. Okay. We can't produce the protein without the mRNA, okay? Because that's our instruction manual. Okay. So that's the mRNA. The second thing that we're going to need are some ribosomes. Okay. Then we're going to need some tRNA molecules. Okay. And then the last thing that we're going to need are some enzymes. Okay. All right. So we've already talked about the mRNA. Okay, the next thing that we're going to be looking at here is the ribosomes. Okay, so before we get into that, we want to choose all the correct facts about ribosomes. Okay, so there's two answers for this one. So we have A, ribosomes function in glucose synthesis. B, they function in nucleic acid synthesis. Or C, ribosomes function in protein synthesis. Okay, D, ribosomes are found on the smooth endoplasmic reticulum. E, they're found on the rough endoplasmic reticulum. Or F, they're found on the Golgi apparatus. Okay. Which two of these are true? All right, so Paul says C and E. Anyone agree? Anyone disagree? What do we think? Anybody? Oh, okay. Well, Paul is correct. Yeah, so C is one of them. So ribosomes are going to function in protein synthesis. And then E is also correct because they're going to be found on the rough endoplasmic reticulum. Okay. So if you remember when we were back in chapter 3, I believe it was, okay, ribosomes are always going to be found on the rough endoplasmic reticulum. And their job is to be able to help produce proteins here. Okay, so they're going to be the second most important character in this process. Okay. Ribosomes are going to be made up of a second type of RNA called ribosomal RNA or rRNA. Okay. So far we've already seen messenger RNA or mRNA. And there's a second type of RNA called ribosomal RNA or rRNA, and that is what actually helps to produce ribosomes. Okay. So this ribosomal RNA is going to be made inside the nucleolus, okay, within the nucleus here. And so that is going to be what actually forms the ribosomes. Okay. So when we're looking at ribosomes, depending on whether we're looking at a prokaryote or a eukaryote, ribosomes are going to be found in one of two places. Okay. In prokaryotes, ribosomes are only going to be found within the cytoplasm because, again, they're not going to have any organelles like the ER. Okay. In eukaryotes, ribosomes are going to be found on the rough endoplasmic reticulum, and they can also be found on the cytoplasm, but for our sakes, we're just going to worry about them being on the rough endoplasmic reticulum. Okay. So we're going to be looking kind of mostly from the perspective of eukaryotes here for the sake of our discussion today. Okay. So ribosomes are the second major player in this process. Okay. So when we look at ribosomes are made up of two subunits. Okay. We have a large subunit, okay, and then we have a small subunit. Okay. The small subunit is the part of the ribosome that actually binds onto the mRNA that we produce from transcription. Okay. So that's going to be the first portion here. Okay. The large subunit is going to be the larger part, and so the large subunit is actually going to bind to a third type of tRNA molecule. Sorry, a third type of RNA molecule called tRNA. Okay. So the large subunit binds to tRNA. The small subunit binds to mRNA. Okay. And so this is the tRNA molecule over here. Okay. So we had a large subunit and then a small subunit. Okay, so that's going to bring us to our third player, our tRNA. Okay. So the tRNA molecule is our third type of RNA here. And so what it's going to be doing is it's going to be bringing one individual amino acid to help grow our polypeptide chain. Okay. And so this is what one full tRNA molecule looks like here. And what we're going to see here is at the top, we're going to see a bunch of kind of atoms here, a bunch of molecules attached to an amino acid. So each tRNA is going to be responsible for bringing one amino acid into the fold, okay, to help add or help form our polypeptides here. Okay. In order to do that, each tRNA is going to bind to a specific mRNA sequence. Okay. So the tRNA is going to bind onto mRNA, okay, and as it binds, it's going to bring that amino acid and add that to the growing polypeptide chain. And so we'll take a look at what that looks like in a second here. But essentially what tRNA is going to be doing, or transfer RNA, okay, what transfer RNA is going to be doing is it's going to be able to translate the mRNA into an amino acid sequence, okay, to help us know what amino acids to produce or what amino acids to bring at any point in time. Okay. And we'll look at how this is going to work in a little more detail going forward. Okay. So if it doesn't make sense now, don't worry about it. It'll make sense in a bit. So that's tRNA. Okay. Okay, so we've seen mRNA, we've seen the ribosomes, but we've seen the tRNA. Okay. And so one thing that we want to kind of keep in mind here is that, first of all, transcription is going to help us generate the mRNA. Okay. We form our mRNA through that transcription process. Okay. Then what we're going to do now is we're going to take that mRNA, okay, and we're going to use that mRNA to help us produce some proteins through this process of translation. Okay. We're going to use the nucleotides in mRNA to provide the information to let us know what amino acids to bring at any point in time. Okay. So when we're looking at these amino acids, okay, there's about 20 naturally occurring amino acids that just happen to show up in nature. Okay. So about 20. Okay. Now the question is, how do we know which amino acid to bring to grow our polypeptide, to create our protein? Well, what we're going to be doing here is we're going to be looking at the mRNA template, okay, the mRNA that we produced, okay? And we're going to be looking at every three nucleotides, every group of three nucleotides here, okay? So every three nucleotides, three nucleotide mRNA sequence is referred to as one codon, okay? So this AUG, okay, this would be one codon, okay? This is a GUC, so that would be your second codon, okay? Here we have AGU, that would be your third codon, and so on and so forth. Okay. So each codon, okay, each group of three nucleotides is going to code for a single amino acid. Okay. Each codon codes for one amino acid. Okay. And so at the end of this whole process, we can have a polypeptide from anywhere from 3 amino acids long to anywhere to maybe thousands of polypeptides long. Okay? Okay. Again, we have a total of 20 naturally occurring, but they can be found in any number of combinations to help us form our protein. Okay? Now, in order to help us form that protein, we're going to be using all of these codons to help us know which amino acid comes when. Okay? So each codon is going to code for 1 amino acid. Okay? Each group of 3 nucleotides is 1 codon, and each codon codes for 1 amino acid here. Okay? And so the relationship between the codon and the amino acid that it corresponds to, okay, is called the genetic code. Okay? So this is what this whole process is going to look like here. Okay? Oops. So this is the genetic code here. Okay? So we have a total of 64 combinations of codons that we're going to be seeing. Okay? And again, each of these codons, or I guess 61 out of the 64 codons, is going to code for a specific amino acid. Okay? So again, like we said before, we have 20 naturally occurring amino acids, but we have 64 total combinations of codons. So we're going to see some repeats here. Okay? So for example, for the amino acid, arginine, okay, we can have CGU, CGC, CGA, CGG, or AGA and AGG. Okay, these are all 6 different codons, 1, 2, 3, 4, 5, 6, that all code for the same amino acid. Okay? But each codon is going to code for a specific amino acid here. Okay? So there's going to be some repeats. Here, for example, we're looking at the amino acid threonine. Okay? That's going to be formed by the codons ACU, ACC, ACA, or ACG. Okay? All 4 of these can help form the amino acid, or help code for the amino acid threonine here. Okay? So in order to begin translation, in order to begin the whole process of producing a protein here, okay, we're going to start at a specific codon. Okay? That codon is called the start codon, and that will always be the codon AUG. Okay? AUG is always going to be where translation begins. Okay? It doesn't matter if you have a bunch of nucleotides before you hit that AUG, they don't count. Okay? You only start at the AUG. Okay? So that's going to set your reading frame. That's going to tell you where you're going to start translation. Okay? So this AUG, that's going to be your start codon. Okay? And so this will always also code for the amino acid methionine, or MET. Okay? So once you have your start codon, then again, you go through every 3 nucleotides, you reach your next codon, you add the next amino acid, you hit the next codon and add the next amino acid, hit the next codon and add the next amino acid, and so on and so forth, okay, until you reach one of 3 stop codons. Okay? So there's 3 stop codons. These are the UAA, UAG, or UGA. Okay? So these 3 codons here, okay, are going to tell you when to stop translation, okay, when you're going to stop your protein synthesis. Okay? These do not code for any amino acids. Okay? All they do is they tell you when you're done. Okay? So the amino acid that comes before the stop codon is the actual last amino acid in the chain. Okay? So the way you can kind of remember this whole process here, or what these 3 amino acids, or these 3 codons here are, okay, is UAA for you are away, UAG, you are gone, or UGA, you go away. Okay? Those are the 3 stop codons. Okay? UAA, UAG, UGA. Those tell you when to stop, okay, when to cut your protein. Okay? Those do not code for any amino acids. Okay? So for example, I'm going to go ahead and come back to the previous slide here. So here we saw 1 codon, we saw another codon, a 3rd one, a 4th one, and then we see our UAA, which is our stop codon. Okay? This one does not code for an amino acid. That just tells you when you're done. Okay? So the amino acid that came before the stop codon is the last one in the chain. Okay. Okay. Any questions so far? Is this making sense? I know it's kind of a lot going on here, but any questions here? Okay. Okay, cool. So we'll go through a few more examples, and this will make a little bit more sense here. Okay. Okay. So, like I said, this is the genetic code, and then I have a table over here that tells you the full name of each of the amino acids. Okay. So, and then their corresponding 3-letter abbreviations. Okay. So, for example, arginine is ARG. Okay, that's going to be this guy over here. Asparagine is ASN, which is over here, and so on and so forth. Okay, so these are the full names of the amino acids that correspond to their 3-letter abbreviations. Okay. Okay, so translation is going to take place in 3 stages, just like transcription did. Okay, we're going to see an initiation stage, okay, then we're going to see an elongation stage, and then we're going to see a termination stage. Okay, and this process is going to work exactly the same way in prokaryotes as it will in eukaryotes. Okay, so the first process is initiation, and so in initiation, what we're going to do to start our translation process is we're going to begin by forming an initiation complex. Okay. So the initiation complex is going to be formed by the small ribosomal subunit. Okay. We're going to have the mRNA template. Okay. We're going to have 3 initiation factors, which are just enzymes involved with this process. You don't need to worry about what those are. Okay. And then we're going to have an initiator tRNA. Okay. Remember that tRNA is going to be bringing an amino acid. Okay. The initiator tRNA will always react and always interact with the start codon, which is AUG. Okay? And so it's always going to be bringing the amino acid methionine, our MET. Okay? So to start the whole process, we bring our small subunit of the ribosome, we bring the mRNA, some initiation factors, and then an initiator tRNA, which will bind onto the start codon of the mRNA here. Then once we've done that, now we're going to begin the elongation phase. Okay. So in the elongation phase, what we're going to have is our large subunit, our large ribosomal subunit is going to come in. Okay. Now when we look at the large ribosomal subunit, okay, there's going to be 3 compartments in there. Okay. There's going to be an A site, okay, there's going to be a P site, and there's going to be an E site. The A site is where we're going to have a tRNA molecule that is charged enter. Okay, so when we're talking about a charged tRNA molecule, all that means is that it's a tRNA that is bound to an amino acid that it's going to be bringing in. Okay, so the A site is going to be where the tRNA will enter the ribosome from. Okay, then we're going to have a P site here, and that P site is going to be where we have our tRNA that is attached to the growing polypeptide. Okay, so P for polypeptide. Okay, P is going to be where we find our growing polypeptide at. Okay. Now again, this is going to be where our tRNA is going to be bound to the polypeptide. It hasn't yet detached from the tRNA just yet. Okay. Then we're going to have an E site. Okay, and the E site is going to be where the tRNA is going to get ejected out of the ribosome from. Okay. So when that tRNA attaches its amino acid, it's eventually going to dissociate from it. Once it dissociates, it's going to enter the E site, and then it's going to exit out of the ribosome from the E site. Okay. Okay. So again, I know it's kind of a lot going on, but we're just kind of introducing all the players, and we'll see how this whole process is going to work. Okay. Okay. So that's what's going on with the ribosome. Okay. Now, also what's going to be happening here is that the tRNAs here are going to be having a specific region that's going to bind onto the codon. Okay. So each tRNA molecule is going to have what's called an anticodon, okay, that helps it to bind with the mRNA's codon. Okay. The anticodon is going to be complementary to the codon of the mRNA. Okay, so for example, here the codon is GCC in mRNA. Okay, so the corresponding anticodon in tRNA would be CGG. Okay, complementary to these 3 bases here. So each tRNA is going to have its own anticodon, okay? And that is going to be what attaches to the codon and bring the correct amino acid into the polypeptide here. Okay. So that's what the tRNA molecule is going to be looking like there. Okay. So now here's how this whole process of adding these nucleotides is going, or adding these amino acids is going to go. Okay. The ribosome is going to start at the AUG, starting at the start codon, okay, and it's going to move 1 codon at a time. Okay, every 3 nucleotides is going to shift over. Okay, so it's always going to be shifting over 1 codon at a time. Once it shifts, it's going to add a tRNA that is charged. The tRNA is going to enter the ribosome at the A site, okay, which we see over here. Okay, once it enters the A site, it's going to add its polypeptide, or it's going to add its amino acid to the growing polypeptide here. Okay, that's what we see here. So it enters at the A site. Once it enters, okay, then you're going to attach that amino acid to the growing polypeptide that's already present. Okay. The next thing that's going to happen is we're going to see that the polypeptide is going to detach from the tRNA that's in the P site, okay, and it's going to be latched onto the one that's in the A site. Okay. The tRNA that is in the P site is going to end up moving to the E site and then getting pushed out of the ribosome, getting ejected out, okay, while the one in the A site takes its place in the P site, okay? And then you start this whole process all over again, okay? So just to kind of recap here, okay, we're gonna have our tRNA that's going to enter at the A site, okay? tRNA enters at the A site and it's going to attach its amino acid to the growing polypeptide here, okay? Once it attaches, okay, we're going to have now an uncharged tRNA, one that no longer has its tRNA attached. Okay? It's going to move into the E site. The one in the A site is going to take its place in the P site. Okay? The tRNA that moves into the E site is going to get ejected out. And the one in the P site is now going to be ready for a new amino acid to attach to it when a new tRNA comes in at the A site here. Okay? So the tRNA that gets ejected from the E site is eventually going to get recharged, and it's going to have a new amino acid attached to it. Okay. All right, so this is just kind of a few big pictures to kind of help you kind of see this whole process a little bit easier. Again, you have a tRNA molecule that is charged entering at the A site. Once it enters, okay, you're going to attach the growing polypeptide to the amino acid, to the new amino acid here. Okay. Okay. So now they've been attached at the tRNA that's now in the A site. The one in the P site is now uncharged. It's going to move into the E site and get ejected out. The one in the A site takes its place in the P site, and now you're ready to start this whole process over again, okay? Over and over and over until you're done. Okay. Okay. Any questions on this process so far? Okay. And again, we'll look at this in more detail in class next week. But does this make sense? Are there any questions on this process here? Okay. All righty. Okay, so here we have a question. Okay, so we have an RNA sequence: AUG, CCG, AAA, CGU. Okay, so which of the following statements describes how the RNA sequence specifies the production of an amino acid chain? Okay, so how do we know how we're going to be forming our amino acids here? Okay, so we have A: each set of 3 bases codes for a single amino acid. B: each group of 3 bases codes for an enzyme that binds amino acids together. C: each base produces electrical signals sent to the cytoplasm to initiate protein assembly. Or D: each base codes for 1 amino acid. Okay. It was a little bit of a tricky one, but I think you guys got it. What do we think the answer is? How do we know how we're going to be adding our amino acids here? What do we think? So Paul's saying A. A is in fact the correct answer. Okay, so this is going to be A. Okay, so remember, each set of your 3 bases is going to code for 1. Okay, each 3 bases codes for 1 codon, and each codon is going to bring 1 single amino acid. Okay, so every 3 bases is going to code for 1 amino acid. Okay, 3 nucleotide bases comprises 1 codon, and 1 codon will give you 1 amino acid. Okay. So A is the correct answer here. Okay. Any questions on this one? I know this is a little bit of a tricky one, kind of weirdly worded here, but this one makes sense. Are we okay with this? Alrighty. Okay, looks like we're okay. Okay. All right, so that is how we will eventually start growing our polypeptide, okay? 1 amino acid at a time. So the ribosome is going to shift 1 codon at a time, and as it shifts, it's going to add a new amino acid. Okay, as it shifts another one, it's going to add a new amino acid. As it shifts, it's going to add another one, add a new amino acid. So you're going to continue elongating your protein. Okay. Now, eventually we want to stop producing our protein, and so we're going to have this last phase called the termination phase. Okay? The termination phase is going to begin once you reach one of these stop codons, okay? Either UAA, UGA, or UAG. Okay? Once the ribosome hits one of these stop codons, what's going to happen is the polypeptide that we produce is going to get released. Okay? It's going to release, it's going to fold, it's going to become its own protein, and it's going to start doing its own thing. It's going to start doing its whatever function it's designed to do. Okay? So you're going to release your polypeptide. Okay? The second thing that's going to happen is we're going to break down our ribosomal subunits. Okay? So here our subunits are kind of oriented together. Okay? At this process here during termination, we're going to dissociate the two ribosomal subunits, and they're going to detach from each other. Okay? And they're going to leave the mRNA. Okay? The last thing that's going to happen is the mRNA is going to end up getting degraded. Okay? So you hit a stop codon. Once you hit the stop codon, you release your polypeptide, you break down the ribosome, and then you start to break down the mRNA. Okay, cool. Okay, so now we're going to go back to our translation activity here. So here we go here. Okay, so I'm going to go ahead and just kind of speed through this here. Okay, is everyone able to see the activity again? Yeah, okay, cool. Okay. So here we're going to begin making our protein. Okay? And so here at the bottom, we have our mRNA nucleotide. So this is our codon. Okay, we have 1, 2, 3 nucleotides in our mRNA. This is our single codon here. Okay, and so remember that we're going to be bringing in a tRNA molecule that is going to be bound to a specific amino acid. Okay, and that tRNA molecule is going to have an anticodon that is going to be complementary to the mRNA's codon. Okay, so which of these four has an anticodon that is complementary to this GGA over here? CCU, perfect. So we're gonna have CCU, and that's gonna bring the amino acid glycine. Okay, now we have a GCU. Okay, so what's the corresponding anticodon? And what amino acid are we gonna be bringing? All right, so what's our corresponding anticodon? CGA, right. So we're gonna have a C, which is gonna correspond or complement the G. We're gonna have this G, which is gonna complement this C here, and we're going to have this A, which is gonna complement this U here. Okay. So this is going to be the next complementary, or sorry, the next anticodon on the tRNA. So this tRNA is gonna bring the amino acid alanine. Perfect. Good. Okay. Now we have our next codon, which says GCU. Okay. So which amino acid or which tRNA molecule is going to be complementary to this GCU? CGA, perfect. Right, so we should have CGA, which again is going to bring in our alanine amino acid. Perfect. Okay, and again, you can start to see our polypeptide is beginning to be formed here inside of our ribosome. Okay, next one here is CG or CUG. Okay, so it'd be the corresponding anticodon. That would be GAC, which carries leucine. Perfect. Cool. Good. Okay. And then again, what you can kind of see is you have an incoming incoming tRNA. Okay, that's going to be adding its amino acid here. Here's the E site. Okay, and so all of these tRNAs are going to be uncharged and getting pushed out. So here we have UCU. And so Brianna says we're going to be bringing in serine. So AGA, that's perfect. Exactly. So we're going to bring in the amino acid serine. Okay. Good. Okay. Here we have the codon ACG. Okay, so what would be the corresponding anticodon? And what amino acid are we bringing? Oh, that's all good. Yeah, so we have UGC, right? UGC, so that's going to be our threonine here. Perfect. Good. Okay. Now we have AUU. Okay, that's our codon. What's our corresponding anticodon? UAA, perfect, which is isoleucine, perfect. Okay. Now we have CUG. So what are we looking for here? Right, we have GAC, which carries leucine. Good. Okay, now we have CAA. So we need GUU, perfect. Good job, Brianna. Cool. And now we have AGC. Should be UCG, perfect. So that would carry serine. Good, good job, good job. Okay, now we have UUU, which would bring us which anticodon? AAA, perfect. So that would bring us phenylalanine, perfect. Okay. All right, so we formed our protein here. Okay. And so the next thing that's going to happen is we're going to continue building our tRNA, or sorry, building our protein here until, whoops, let me, okay. You're going to continue building your protein until you reach one of these stop codons. Okay, here we have UAA, which is our stop codon, and that's going to tell us when we're done. Okay, that's going to tell us when we're completed, when we've completed reaching our protein. Okay, we're going to detach our protein. Our protein is going to go off and start functioning the way it's supposed to function. Cool. Okay, so that is that. Okie dokie. So let me go back here. All right. Okay. Any questions on translation at this point? Is everyone able to see the PowerPoint now? Okay. So all making sense so far. Cool. Okay. All right. So that is translation. So we have just formed our protein. Okay. So quick question here. Okay. So we have an RNA sequence that is 12 bases long. Okay, so what is the maximum number of amino acids that can be coded for by this sequence? Okay, if we have 12 bases long, okay, how many amino acids can we produce here? Okay, is it 12, 3, 4, 6, or 36? What do we think? Right, so the answer would be 4. Perfect, right, because you have 12 amino acids. Okay, so I'll just kind of draw out. So here's one. I'll just kind of draw these little lines here. Those are going to represent your RNA nucleotides. I lost count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, right? So these are all of our nucleotides. And so every 3 is going to code for 1 codon. Okay, so that would be 1. Here's a second codon. Okay. Here is a third codon, and then here's a fourth codon. Okay. So because we have 4 codons that are made up by these 12 amino acids, okay, we're going to end up being able to form a maximum of 4 amino acids here. Okay. Good. Okay, that's good. Okay, so next question. So let's say we have a deletion mutation. So a deletion mutation occurs, resulting in an RNA sequence that is 11 bases long. Okay, so what is the maximum number of amino acids that can be coded for by this sequence? Okay, 11, 6, 4, or 3? What do we think? Right, 3 is exactly right. Perfect. So we're only going to be able to form a maximum of 3. And again, the reason for that is, again, if you're starting with 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 amino acids. Okay. Now we start to look for our codon. So here we have 1 codon here, 1 group of 3. Here you have a second codon, which makes up another group of 3. Here's a third codon, which makes up another group of 3. And then we have just 2 left over. Okay. And so because we have 2, we don't have another codon. Okay. And so the maximum number that we can come up with is 3 amino acids here. Perfect. All right, good. Good job. Good job. Good job. Okay, cool. All right, so now we have an RNA sequence here. Okay. And so here we have our RNA sequence, which is ACG, AAA, GAU. Okay. So using the codon chart, what is the sequence of amino acids that is produced when this gene is translated? Okay. So before we answer this question, there's one thing I want to bring our attention to because I forgot to mention it earlier. Okay, so when you're looking at the amino acids that we're going to be producing, there's 2 ways of determining what your amino acid sequence is going to be. Okay, option A is you can look at the anticodons. Okay, and that's assuming you have the tRNAs with the proteins, okay, or the amino acids, or you can take a look at the chart, okay, the genetic code chart. Okay, this is option B, and this is going to be the one that we're going to be using most often for our purposes. Okay, so using our chart here, try to figure out what you think the amino acid sequence would be, okay, using our RNA sequence here and the corresponding amino acids. Okay, let me know what you guys think the answer is. So this might take a little bit. What are we thinking? Okay, so I'm seeing A's, yes, and A is absolutely correct, right? So threonine, lysine, and then aspartic acid. So, and the way you do this is you look at the first codon, ACG, and we look for ACG here, and we see ACG is right here, right? So ACG would code for threonine, perfect. Then we have our AAA. Our AAA would give us lysine here. Okay. And then we have GAU, okay, which is over here. GAU, which gives us aspartic acid. Okay. So that would be the amino acid sequence from this particular RNA sequence here. Perfect. Good. All right, and that is that. What questions do we have about? What questions do we have about transcription or translation? Are there any questions here? Okay, so if there's no questions, I'll go ahead and show you guys what you'll be doing for lab today. And so this lab is going to be relatively short. This one I pulled straight out of the lab manual, okay? And so we're just gonna be doing activity 2, okay? And so what we're gonna be doing is, the first thing you're gonna do is you're going to look at this website. And so that was the website where we just did that transcription and translation simulation. So Mary's questions for the lab, question 3D, it starts with AUG. Yes, and so I'll talk about that in a second. Yeah, so hang tight real quick. Okay. So first thing you're gonna do is you're gonna go through this website, okay? And I want you to actually watch the videos and then go through the simulations to help you practice transcription and translation. Okay. After you've done that, here we've got a process or 2 DNA sequences, okay? Complementary strands. So here we have 1 strand of DNA across the top, and then across the bottom we have another strand of DNA. Okay. So what we're going to be doing is you're going to be creating an mRNA sequence using the top strand as your template strand. Okay. So this top strand is going to be your template. Okay. And you're going to produce an mRNA sequence, and you're going to write that in here. Okay. Okay, so that's going to be your mRNA. Okay, that's going to be your transcription process. Okay. Now, once you've formed your mRNA template, the next thing you're going to do is you're going to translate that mRNA. Okay, so whatever mRNA template you come up with here, okay, you're going to copy that, drop it into this, this box over here where you have mRNA again. Okay, and then you're going to underline where you find the first AUG. Okay, because again, remember the AUG is going to act as the start codon. Okay, that's going to be where you're going to begin translation. Okay, regardless of whatever comes before that. Okay, okay, so that's going to be where translation begins. Okay, so you're going to underline wherever you find that first AUG. Okay, and then you're going to underline every 3 bases. Okay, okay. Once you identify those 3 bases, or each of the individual codons, you're going to translate that mRNA and list the proteins, okay, that you're going to be forming, or sorry, the amino acid that you're going to be forming. Okay, so you're going to list the amino acid sequence, and you're going to use this chart here, okay, to help you identify what amino acid comes when. Okay, so using your mRNA, you're going to identify what your amino acid sequence is going to be here in the protein box. Okay. And again, you're going to start translating at AUG. Yes, absolutely. You're going to start translating at AUG. Cool. And because that is the start codon. Okay. Okay. So once you do that, then there's just a bunch of questions here. So the first question asks how many amino acids are in your protein. The next one asks about what the corresponding anticodon is for AUG. Next one asks for the anticodon of being CA. So what would be the corresponding amino acid? And you can use the corresponding codon to help you figure that one out. Okay. And then you're just going to answer these questions down here. Okay. That's all you're going to be doing for this lab. Okay. So use what I have on Microsoft Word. Don't use the lab manual because there is an error in the lab manual inside the DNA template, and so it's going to screw up everything. So just use what I have listed here. Okay? Don't worry about using the lab manual. Just use whatever is listed on this assignment sheet that's on Canvas too. Cool. Okay. Any questions on what we're going to be doing for lab? Again, all we're doing is just activity 2. Once you're done with that, you're done for lab, and that's it. Okay. Any questions on what's happening here?